recording. And another thing, there are two of me, so my name is twice, but I didn't clone myself. I'm just having some Wi-Fi connection error difficulties. So you're hearing me from my phone. I hope you can hear me all well. And I hope that even if my screen is frozen, you still hear me and can just shout out, then I can just make a pause and then start over. Sorry for this, but it's the only way to do this. So you're not seeing the right screen, are you? Ah, you are. Great.
Yep,
okay.
yeah.
Great. So welcome everybody. I'm happy that you all joined. We are going to discuss the STURDY trial. So I love the acronym, so that's the reason I had to write it out. The study to understand SAL reduction and vitamin D in youth. And we are going to use the results paper, which is the one which was the required reading for today. It was published a year ago in Annals of Internal Medicine. And as Dave Shade mentioned, we are very lucky that within our track there are some people who have worked on this trial, and I was very lucky that I could get some background information, and also very lucky that we have even more background information joined here today. Thank you for this. So, but to come to the fun part, we are going to play some bingo today, or at least the students will play some bingo. And they're just going through the bingo instructions because I'm not calling out any numbers. It's actually you who will decide if you can mark a field or not. So every student who is in the track and who's presenting should have a card. I hope everybody has opened their card. Let me just check, for example, Miles, could you open your card? Could you just shout out? Sorry to call you out, but I'm just seeing your name here.
My card is working okay.
Great, perfect. Thank you, Daisy. So, and just the instructions. So how can you mark your field? So we'll get some questions during the journal club, which aren't related to chocolate, but here for the example, I'm asking where does the best chocolate come from? And let's say somebody is saying Switzerland. Then if you say Switzerland, you can mark your best chocolate label on the bingo card. If some other student is saying Belgium, they can mark their card as well. So it's not first come, first serve. If there are many answers, you can mark your bingo card as well. And so we go through the presentation, and I'll ask another question. Let's say, well, does the best coffee come from? Somebody says Colombia, and the person can mark the best coffee field, and so on. Best porcelain, China, mark your field, and you can win if you have a line, so one horizontally, one vertical or diagonal full field. Your cards have 4 fields, so it's here there are 3, you have to have 4. Or the winner is the second winner or the first winner, depending on the situation, is the one with the most answers. So I'm talking about winning. So what are you gonna win? This is your prize. It's original Swiss chocolate I brought from Switzerland. So it's worth participating today. And to make it a bit easier for you students, you have some special rules today. You have a joker. It's a faculty joker. So what does it mean? For example, there's a question and you see it's really important on your bingo card, but you have like no idea what to sell. Then you can just chime in, say, I want a faculty member to answer the question for me. Then you choose one faculty member. So for example, I say, Stefan Erhard, could you please answer this question for me? And then poor Stefan has to answer the question for me, but I can mark the question and get the point. And there is some special field. It's called discussion, and it's just if you're participating without any specific topics. So we are going to talk about. Sorry. about some things, and if you're just participating in the discussion, you'll get the feel for this. So somebody just asked me to get a bingo card. That's the reason I'm just stopping for a second here. That's the moment where you all can chime in and tell me if you need another bingo card. I think you asked me, I'll post one link. It's just for her. If anybody needs another, I'll post another, but please use, so that's the next one, if anybody wants to join. Okay. Any questions related to the bingo? Silence. Okay, so I'm proceeding. So some background information. I'll keep it short and simple. We are going to talk about falls in older people, and it's a very important topic. Just to name some of the numbers from the publication, there are about 2.8 million older adults treated in the emergency departments after falls, and over 800,000 are hospitalized after a fall. And maybe to stay with my statement, to keep it short and simple, I'll just quote one quote from a book. Actually, it's from Love in the Time of Cholera. There, the author stated, "Old age starts with the first fall and death comes with the second." And I think this summarizes really great the importance of falls in elderly. And if you have any questions about background, I think we can swift it to the end of the discussion. But I think this is emphasizing the importance of falls in elderly people. And when we're talking about the problem, we always want to have a solution. And so one idea was to treat patients with vitamin D and then to decrease their risk rate and to then decrease all the related morbidity and mortality coming from the falls. But before the study trial, actually, there was some equipoise in the literature, because there was some studies saying that the fall risk can be reduced by 20%. There were other studies who said the fall risk is increased and no effects, and all the literature were using different dosages, so we didn't know where to go. So that's the reason the study trial was started. So, which persons were included in the trial? First bingo question, anybody? Just unmute yourself and shout out, or write in the chat. I'll try to monitor this as well.
Elderly.
Elderly, exactly. That's one bingo label for you. Jasmine was said age over 70 years. Exactly. Jasmine, you can mark your field as well.
And they have to have an elevated fall risk and also low serum.
Elevated.
Yeah, low serum vitamin D level. Sorry, did you hear me?
Yeah, that's. Yeah, I hear you very well, Ling. You can mark your field as well. Anything? I think we got everything. So the only thing I took it's their community dwelling adults. So not hospitalized, not in retirement homes, so they're home alone, so a bit more fitter population here. And you mentioned the elevated fall risk, or let's start with the exclusion criteria. Not a bingo question. So cognitive impairment, hypercholesterolemia, some kidney or bladder stones, and when they're using vitamin D above the dosages of the trial, they can't be included. And we were talking about the elevated fall risk. Can anybody name the author's definition about elevated fall risk? How did they define this risk?
I can just read it. It's a self-reported one or more of the following: 2 falls or 1 injury injuries for in the past year, fear of falling due to balance or walking problems, difficulty in maintaining balance, or use of a assistive device when walking.
Exactly, you named all of them, so you got all the answers, Ling, just for you to mark the bingo field. So the person had to have 2 falls, 1 injurious fall, or has some balance walking, difficulty maintaining balance, or has to use an assistive device. When I saw this list, my first impression was this might be a bit heterogeneous risk assessment. So I had the impression maybe to fall, if a person was falling 2 times, it's the person has a higher risk compared to somebody who's just using a assistive device. Just a hint for the discussion, or to look out in the results for this. So, but the next, the recruitment, just and the original paper, there was written that it was mass mailing of the brochures, but in the protocol or the design paper, I was really impressed, and I think it's really important and cool that they had so many other strategies. So just want to list them. They had 7 other options if their first option of mass mailing the brochures would fail, and I think this is amazing plan to keep to have ideas if your recruitment is failing. So going to the next. So we have the study has different trial phases, so the dose finding phase and the confirmatory phase. And just as an exercise for your comps or the CTS track students, can anybody tell me what type or how would you describe this type of design? If you're just looking at the dose finding phase, if this would be the trial, how would you name it?
This is a parallel.
Yes, that's one right answer. Anybody else? Jasmine, responsive adaptive trial. Yeah, Jasmine, that's if maybe if you're looking just at this phase, it can be adaptive as well, yes, but you're going into detail. Is there anything else, for example, about the way the treatment and the control is mentioned or is placed? Changji, sorry to call you out, but any idea what we have missed here saying? So we said.
Well, I would say this is multi-arm, 4-arm trial, and also it is a dose finding trial. That's the 2 things that I could think of.
Perfect. Yeah. Thank you. If you're playing bingo, mark your bingo field for this. Just some of my thoughts, or I was thinking, so it's individual participant randomized to a multi-arm parallel superiority trial with an active control. So maybe this would be the way I would describe this phase of the trial. And if we're looking at the
I'm sorry, I was just going to add also, you might add masked or blinded to that list of descriptors.
Yes, and since you brought it up, any of the students can say if it's masked or blinded trial. So just because it was blinded, so the participant and also the centers were blinded to the treatment. And we'll talk about this blinding issue in a minute as well. So to repeat the exercise for your comps, how would you classify the confirmatory phase of the trial if you want to name this trial? Morgan, do you want to give it a try?
Can you repeat the question, please? I'm sorry.
No worries. How would you call this kind of a trial? So about the design.
Dose response, possibly.
Yeah, and about, for example, how the arms are structured. Is it crossover? Is it? So when you're looking just at this page.
I'm not too sure.
So no worries. I would say, again, individual participant randomized, again, to a parallel trial. We want to have this superiority tested, and they're having an active control. That's the way I would describe it. Again, if we think about the comment of day shape, again, here, participant and the centers were masked. So, and when we're going here to this and looking at this result or the trial as a whole, how could you name it? I'll shout out to Jasmine because she was going into this direction. And she can easily mark her bingo field if she repeats her answer. Ling was faster. Yes, it's an adaptive design. Exactly. So what is an adaptive trial? So I had always a bit difficulties to think about it. So I want to use the opportunity to go through this again with you. And if you have any questions, just shout out. I searched for a definition of an adaptive trial, and I found the FDA guidance document for adaptive design clinical trials. And they're saying that an adaptive design is defined as a clinical trial design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. And if you're using Wikipedia, which is also a nice way to just get an idea if you're not sure what it is, it's basically the same. So it's again adapting. An adaptive clinical trial is a clinical trial that evaluates something and has modifying parameters based on those observations. And these adaptations are continuously through the trial, but are prescribed in the trial protocol. And that's the important thing. So you had a plan, you want to change this or that according to something. And you think here, which adaptation can you make? So you have to make a plan. So any idea what you can adapt based on what and why?
New
So
treatments.
very open question.
New treatments.
Sorry, I did.
I just said new treatments that are coming in.
Yeah, great, Camille, that's right, yes.
Also, can we do it on the
Any,
basis of the adverse reactions that we are observing?
Yeah, for
Okay.
example, you can stop the trial if you have adverse events. It actually should be like this if it is in the DSMV. Anything else you could, if you want to stop an arm, do you think of any other reason than adverse events to stop one or the other arm?
Futility.
Say it again, sorry.
Futility. Like we did an interim analysis and we find that the drug is not effective any longer.
Yeah, exactly. Or the opposite, you found that some of the other treatments are more effective, so going in the other direction. Any other ideas what you can change instead of dropping arms or treatments?
Sample size.
Yeah, exactly, Camille. So sample size. So you can go in both directions. Let's say you have observed less events as expected, then you would need a bigger sample size. But let's say your expectations were way too low and you're observing more events, so you can also decrease the sample size. Anything else? Any idea?
Is it possible to change the inclusion criteria at some point? You know, if you found a certain age or certain populations are just having higher risk with the adverse effect?
So I haven't heard about changing the inclusion criteria, but maybe the faculty here can chime in. Any experience about changing the inclusion criteria in an adaptive trial design?
Yeah, please remember it's pre-planned, right? So the scenario that was brought up was probably a reaction to some problem recruiting. So whatever you do, you want to pre-plan this.
So just if you have in mind that you might have troubles recruiting very old people, you might have included it in your protocol to lower the inclusion rate, something like this?
Yeah, so I have not come across that scenario, but perhaps others here have.
I've not worked on a trial where we prospectively planned to change inclusion criteria, even if we, identified particular characteristics in advance. But almost every trial changes inclusion criteria as a result of things you didn't plan.
Good. So just to give you my thoughts about it, basically covered. So we have the sample size adaptation based on the rates, on the variability or adherence. Change treatments, that's what Hermine said, so drop or add is also an option. You can pre-plan. Then you can transition from one phase into the other. And one special case of an adaptive trial design, which we're actually also going to see here, is adaptive randomization. And can anybody name what date is in this study? So in this paper. Yeah, Ling chatted that they changed the treatment. So exactly, they dropped the treatment, transitioning from the dose finding phase to the confirmatory phase. And what did they do? And Jasmine is asking, did they drop some of the dosages? Yes, exactly. They did drop some of the dosages. So both of you can mark your bingo field. And just one other thing, why I actually kind of said it, we're going to talk about this. Adjust randomization probability. Thank you, Shenji. Exactly, you can mark your field as well. So they had these two things. So we're talking now about the patients. Let's just go through one of the patient's flow in brief, and I just want to highlight some of the parts there. So there's an older patient who's has an increased fall risk. There's some screening visits, baseline data will be obtained, and so on and so on. And then there is a run-in phase, and then the person is taking the study pills. That's basically what the study is for the patient. And he's taking the study pills during the dose finding phase and during the confirmatory phase. So that's the schedule. Let's just focus on the run-in. So we had a run-in for 10 days, and there the patient had to take placebo pills, had a daily fall calendar and study pill calendar. So we're talking about run-in. Any idea which type of runnage can be used? So here they use some type. Any idea what I'm referring to? Nakul, maybe you? Any idea?
Could it be
Can anyone help?
compliance?
Yeah, you're shooting for why they're using it, but what did they use? So did they use?
The placebo. I'm not sure. Oh, placebo.
Yeah, perfect. Yeah, exactly. Kamini said it. That's what I'm shooting for. And also Daisy said it in the chat. Both of you can mark your failed. So Daisy is saying in the chat active treatment and placebo. And actually, that is what I'm going for. So here they use placebo, but you can also use your active treatment. Or if you remember, they gave the person placebo, but they could also just ask the person to make a diary. So to see if they can fulfill the diary criteria, so they don't have an actual treatment, but they have an assignment in a run-in phase. And what type did they use here in this trial? To repeat it?
Placebo.
Exactly. Another bingo field for you. Nobody's shouting bingo. It's exciting. So I'm waiting for the first bingo. So here we are coming to the reasons. Kamini was already talking about why. Any idea why to use a run-in phase and which type of these types I've mentioned you can use for which kind of reason? So Kamini, if you want to test adherence, what type would you choose?
If I want to make sure that my patient is going to stay compliant for the rest of the study period, I would go with placebo.
So my internet was cutting out. I don't know if you can hear me, but Kamini, I think you say you're going for placebo. Yeah, you can also go for active treatment if you're trying to test adherence. Any ideas? Any other ideas?
To decrease the dropouts?
Yeah, decrease of dropouts. So you're pre-selecting your patients to the more adherent one. Yes, also correct. Any other ideas?
I may be wrong, but I think researchers can test safety during the running phase. So in this case, they may use active control.
Yeah, right. And just some ideas of mine. So we have to test the adherence. There you can use whatever test you like, at least in my opinion. Then if you had a medication and you are discontinuing it, but it has a long duration until it's washed out, so you can use the run-in phase to wash out this medication. Therefore, you could also use any type of these run-in treatments. If you want to see some side effects, then you should use your active treatments. Otherwise, it would be difficult to see some side effects. And if, for example, you have a drug and it's very important to establish a balance or stable concentration or something like this, then you can use also your active treatment within a run-in phase. And with that, can anybody repeat it? Why do you think that they use it in this study trial, the run-in phase?
I think because they need data for the daily fall and study pill calendar to be filled, so they need to test if the participants can and complete that in retrial.
Exactly. So Daisy and Ling simultaneously test adherence. Both are right. You can mark your sturdy run-in label field. So they're testing adherence. So since I think said it three times that this is very important. Do you think of any other factors which could influence adherence in a trial? So this is a huge list. Just shout out anything you think of.
I think the study length.
Yeah, exactly. Study duration. You're right. Again, I feel for you.
And the time they need to come back for follow-up.
Yeah, maybe the time. So how complex, for example, this study is exactly. Kamini saying if there maybe the new treatments or side effects. New treatments, you think like if a person is seeing a different option, or what do you mean with new treatments?
Yeah, I mean, if they feel that some other alternative treatment that has come up is better, has been told to them by someone that it's better, then they'll start taking on that. Also, if sometimes patients, they stop taking a drug after they start feeling better and they don't complete the entire treatment schedule.
Exactly. And in the chat, Young said convenience. Exactly. So also a bit logistic or simplicity, and also Ling is going in the same direction, numbers of tasks required for them. Exactly. I think you almost covered everything. I was thinking about a few more things. So as mentioned, the run-in phase, so study duration, the setting, where is the location, which intervention, how simple is it to perform it? But there's also participant factors. So if the person believes in the treatment, so has positive health beliefs, it's more likely to adhere. People with mental health issues are less likely to adhere. The logistic factors, there are some incentives incentives could increase the adherence. And there is also some data suggesting that if the participant is very well informed about the study, they adhere better. So the information provided to the participant is also influenced the adherence. And let's look at the adherence in study, and I think they did a great job. It's amazing what a great adherence they had. So here just see in the supplemental table 3, the highlighted one is the percent of first falls with an interview. So if the person was falling, they should tell this to the trial, and you see a high proportion of people called to tell that they have fallen. And even for the pill, it's really amazing. It's 72 and 75% of the people are taking the pills. And if you look at other adherence, or even at real life data, adherence can drop down to 20%. So 72 and 75 is really amazing. So now we're talking about the run-in phase. Anything? Can anybody think about something which might be problematic about the run-in phase? Any problems, risks? This is a bit more difficult, I have to admit, but maybe somebody of you has an idea.
I'm just guessing if the drug used in the running phase is active treatment, it might need longer washout period before we are using real treatment we want to test. I don't know.
Yeah, I think this is a good idea. I haven't thought about this, and Young is saying increase the time to run the trial. That's for sure, depending on the time or the duration of your run-in. And Ling is also going to losing participants, and maybe I losing participants. What is the consequence of losing participants, except besides that your sample size and so on? So what if you have analyzed your trials? What does it mean if you're losing specific participants? So if you're selecting specific, what threat is there?
Are you thinking about a selection bias?
Yeah, exactly. That's the reason. That's Ling and Jasmine, yeah, almost simultaneously. So if you're selecting your patient, you can have a selection bias, and then you can limit the generalizability of your study. And one other thing which we didn't mention, and I was thinking about, so if you have a different type of between run-in and your active treatment, for example. So if you have a placebo treatment during the run-in phase, and then you change to an active treatment, and the patient is feeling a change, this might be a problem where we could lose the effect of the blinding because the person is feeling the change. So that's also a thing to consider. Okay, so going back to the schedule for the patient, we have they're taking the pills, but we have two different phases of the trial. So what happens to a patient who is either on the best dose without knowing it in the beginning, or the control? What does happen during this transition? So they're taking just the same pills the whole time. And if the patient is not on the best dose, so they're changed to the best dose. Exactly. Yeah, exactly. Ling and Daisy, the same. So they're changed to the best dose when the confirmatory phase is starting. So just one thing we read. Oh, bingo! Great, Daisy. First bingo. Congratulations. So first winner. Now we're just shooting for the most answers. So keep tuned, give the most answers. I have a second chocolate there, so there's still a chance to win it. So just to give you some information, if the patient was changed from having not the best dose to the best dose, you said he was switched, and it was they actually told me this. It was a really logistic problem because you wouldn't want to unblind the participants or the clinical center, so you had to find a way to change the dose without anybody knowing. And so they used this really cool trick. They just didn't transition every patient at the same time, but they waited until they had to refill their container anyway, and then change the dose, which I find a really great trick to do. And just one thing to mention here as well. Anybody can remember what they were saying about the analytic part? What have they done with the people who were not on the best dose while analyzing the paper? So to just shorten it a bit, so they took the time from the time point when they were switched. So the time beforehand didn't count for their confirmatory phase, which is also a great deal to deal with this problem about these two phases. So I promised the adaptive randomization part. I think it's also important for all of the students to get it, because sometimes it's confusing what they're actually doing. So I just go through this example because I think there you can understand it. So in the beginning, you have these three doses and the controls, and the probability to get assigned to the control group is 0.5, and the others are randomized by 0.1667 probability in one of these three dosages. And after the 100th patient was randomized into the control group and completed a follow-up of 6 months, there was an analysis done, and the probability was calculated which for the dose to be the best dose. So with the analysis, the first analysis, dose 1 had the highest probability to be the best dose, and so the probabilities of being randomized to this group changed accordingly to this analysis done. So from now on, after the first analysis, the people were more likely to be randomized to dose 1 and less likely to be randomized to dose 2 and 3. And the amount of people who were randomized to control stayed the same. And just to give you a visual depiction, this was actually, if you're seeing the initial randomization, it was 3-1-1-1, and now it's changing completely. So almost all of the participants are either randomized to the control or to dose 1, and just a few patients are randomized to dose 2 and 3. So, adaptive randomization. On which factor can you base your change? Any idea? What can you use? So you can base it on some prognostic or person factors. For example, something you are collecting within the screening process, some characteristics. Importantly, you have to know it before the randomization to do the randomization, or you can base it on the outcome. So, for example, to base it on data you have gathered after the first patients were randomized to this group. And just one thing, the outcome has, normally it's everywhere written that the outcome has to be known quickly, and maybe it's depending there quickly is depending on what you're aiming for. Here you had a delay of 6 months follow-up, but for example, I was thinking it's more important that you're not waiting for sample to analyze some biospecimen in the end. You can't use this information if you're analyzing it in the end of the trial. So you have to have the information to make the change. And what did they use in study from these two options? So they based it on the outcomes. So and we have a bit of time, and I think maybe this is important. I just want to open the field for some ideas, thoughts about the discussion of pros and cons of the adaptive trial design. I've hinted some of the parts, but I just open the field, and then maybe I can chime in with my ideas. Any thoughts? Faculty is also welcome to give their opinion.
So with adaptive trial design, we make sure that the patients are always getting the best treatment available option. And the the disadvantage I'd say is that you'll have to keep training the staff and the study personnel again and again as to what is changed and how they have to move further with the entire process.
Yeah, exactly. Good point. Just one thing I want to show you, which you can see here in my previous slide, that so this was analysis 1, and the randomization changed dramatically. So there were some pre-specified values you had to, or you were using, and then you had to go with it. So after the 100 patients were randomized to the control group, almost, or just fewer patients, were randomized to the other 2 dosages, and then you have to stick with it. So you're making some assumptions in the planning phase, and then since it's pre-specified, you have to stick with it, and it can change the trial dramatically. So one fault we had while preparing this journal club. Any other idea, opinion?
Oh, Xiangqi, if you have something, go ahead, please.
Yeah, I was just going to say that since the adaptations need to made pre-specified during the trial planning phase, this could add complexities during the trial planning, and also maybe prolong the trial design phase. And yeah, and it may be very difficult to pre-specify things that could be adapted.
Okay, so thank you, Xiangqi. Moving on just to some of the study results. Here's table 1, I think quite balanced. I just want to highlight one thing. I was just seeing while I was reading is the frailty status here. And if you compare it, for example, the control group with every other dosages, I was a bit afraid that there are less frail people here compared to non-frail people in the treatment groups, thinking about the possibility that frail people are more likely to get hospitalized, and if they are falling, have more injurious falls. So thinking about the severity of the consequences there. But I'll show you some subgroup analysis, which then were done and might lower my concerns. Just to go to the outcomes, to have some time. Anyone who wants to tell me which was the primary outcome? So it was first fall and death, and they assessed it very well. Another point I want to highlight, because it's really impressive how they did it. They assessed the falls by a monthly calendar. They had scheduled clinic visits and telephone calls. And as I mentioned before, they had ad hoc telephone contacts after falls. And I think this is amazing, because normally falls are very difficult to capture. And this is the best idea I've seen to capture as much falls as possible. Yes, there is a possibility that still the patients aren't reporting this, but I think they did a great job minimizing it as much as possible. And also death was very well assessed, because they had families or proxies mentioned in the beginning. So if the study team couldn't obtain a calendar from the participant, they knew who to call and to ask about the well-being of the participant. Also very well done. And to just go to, I know I'm running out of time, but just to go through the results, a Kaplan-Meier curve. Some of you are not very familiar with this, so just some quick introduction. Here you have the probability of fall or death, so the primary outcome on the y-axis, and the x-axis is time, and the lines, the different lines are the different treatment groups. And with every step, so when a step occurs, there's an event occurring, and then you're recalculating the probability for your outcome. And you can then see a separation of the different treatment arms. And what is also very important and very well done here is that there is a table stating how many people are contributing to the events, but also how many are contributing to the risk set, because between the events, if somebody is dropping out, they're not contributing to the risk set anymore. So normally this information is giving you more insight about the population who's contributing to this analysis. And you see here that there is some separation, and we were discussing in preparation of the journal club that the confidence interval might overlap in this graph, but they're not shown here. And you see here also in the confirmatory phase, where we have just the control in the 1,000 units, you see almost no difference. And this is also shown here if you're looking at the hazard ratio, where first follow death has a hazard ratio of 0.94, but the confidence interval is overlapping 1. So not a statistically significant difference here. And they also looked at some secondary and safety outcomes as well. Anyone who can quickly name some of them? So gait speed was the secondary outcome, and safety were the rates of adverse events, serious falls, so some with consequences like a fracture, a fall who is then associated with a hospitalization and some serious events. And also there you see that the comparison between the control and the best dose, and you see overlapping hazard ratios. So the confidence intervals are always overlapping. And interestingly, the point estimate is actually favoring the control group, not the treatment group here. Here the other results are going into the same direction, and this one of the subgroup analysis I wanted to show because I had the concern raised with the frail group. And what we're seeing here are some subgroup analysis and also the hazard ratio. So on the right side, more events in the 1,000 units group, so the best dose group, and on the left side, the control group. And what you're basically seeing here is that the confidence intervals are always overlapping 1. So we can't really say that there is a subgroup who might have a total different result compared to the rest of the group. So I ran through the last of the slides, but I really want to give also some, at least 5 minutes to give everyone the opportunity to just talk about something I might miss and they wanted to discuss, or some other insights, some questions. So for this, we have now 4 minutes.
Do you want me to say a few things? Okay.
Yeah.
So here are a couple of, I think interesting aspects of this study. First, the recruitment was just incredible, and that has nothing to do with anyone on this call, I think. I can't see everyone, but the recruiting staff and the design for the recruiting was spectacular. And I wish that all of our trials could recruit as successfully as this one did. But I think it was very labor intensive and very expensive. And I think there's a lesson there, which is that recruitment is always hard and it takes a lot of effort to recruit well in almost all studies. I have not been involved myself in very many studies in which recruitment was easy. Interestingly, this project was part of a request from the NIH to perform a study to examine the effects of vitamin D in preventing falls in the elderly. And they specified that grant applications should design an adaptive trial. And so we worked with a group in the Welch Center to design an adaptive trial, and that was submitted and accepted and funded. But as we continued to refine the design after we were funded, we really began, I think at least many of us, to think that an adaptive design was perhaps not the best way to go about this examination. But the NIH would not let us change the design away from an adaptive one. And so we actually had to follow through with an adaptive design, even though that wasn't perhaps what we thought was best at the end. At the first adaptation, Alexandra showed that the randomization probabilities almost completely eliminated 2 of the 3 treatment arms that were kind of being compared to a control group. That was not what we expected. We would not have anticipated such a dramatic outcome from just the first 100 patients, which illustrates one of the difficulties with doing these adaptive designs. You have to do a lot of work up front to design things very carefully, and then you have to follow the way the algorithm works, even if it doesn't look like it's doing what you expected. Other than a safety concern, you really should not change those criteria. And we found an additional problem related to that, which is that it's not clear exactly why, but the DSMB at one point asked us to remove a treatment arm, even though it was virtually removed because of the low randomization probability, and put us in a position where we weren't sure whether we should retain another treatment arm that was a higher dose than the one they asked us to remove. And that decision also forced us to violate kind of the design principles of an adaptive design to begin with. We were no longer, when we removed one, if we removed either one or both of those arms, we were no longer adhering to the pre-specified adaptive design parameters. So that also put us in an uncomfortable position. We had to figure out how to describe that and in a way that was statistically valid, but also respected the DSMB's role in the process. There's a lot more that we could say. These studies are complex, and I can't speak for any of the other investigators, but this certainly wouldn't be my first choice for a design approach unless the circumstances really, really supported something like this.
Thank you.
Can I ask a quick question?
Any other?
I didn't go into the data analysis that in much detail, but I was wondering in the adaptive design, the study kept the control group at 50% throughout. Is there a reason to do that? Can you actually have more of an equal distribution in the first phase and then switch later?
I think perhaps you can do anything. I saw the primary statistical analyst was on this call before, but she seems to have dropped off. So this is another of the complexities of adaptive designs. I'm going to give you what I think is the answer, but I'm not enough of an expert to know whether it's actually correct. Generally speaking, for just a parallel two-group design, an equal allocation ratio is statistically the most efficient. And one of the reasons you do an adaptive design, there were some others mentioned before, but often an adaptive design is thought to gain you some additional power and or reduce the required sample size. I believe because we were planning to do a dose finding phase and then a confirmatory stage, that keeping everything at a 1-to-1 allocation ratio during that confirmatory stage would have been statistically the most efficient. I'm not sure about that though.
Thank you.
Sure.
Okay.
Well,
Any
so
other last minute?
I think we're done here, Alexandra. I want to thank you for leading a great presentation of a very interesting trial. And I think we might hear about this trial again. It's really a fascinating and successful design and worked out very well. So Alexandra, thank you. Congratulations. Hope everyone is doing well. If you're in Baltimore, you might get some snow tomorrow.